First of all, I would like to thank all the organizers, and especially Michaela, for this really wonderful, very useful meeting and for inviting me. I have a very sore throat, so I'm sorry my voice will kind of break in the middle, but I'll try to do my best. So, as we know that the main ambition for all the patients who once they get a diagnosis is to find a treatment. So at WWGM, uh, we have taken some baby steps in that direction. We haven't achieved a whole lot, but I thought I'll just apprise you of some of the things that we are doing in our organization. So, so the way we look at it is that uh, although, as we have heard yesterday and today, there are really tremendous efforts going on, and we have the possibility of so many treatment options coming up, but we still desperately need treatment options that can slow down and maybe even stop the disease progression. Uh, Now we know that all therapeutic options that have been designed for GNE myopathy or for any genetic disease has its own limitations. So therefore, it's important uh, to make the battle on different fronts simultaneously and try to use different strategies so that either alone or in combination, these strategies may give more benefit to the patients. Uh, and also because GNE myopathy is such a rare disease, so in India and also all over the world, not too many scientists are aware of it. And so it's also a need for us to make scientists interested in our disease so that the research efforts pick up. And the more the research goes on, the more the chances for us to get novel ideas and novel ways to tackle the disease. So, what are the different ways that one can try to get therapeutic options? So we can work at the DNA level uh, because it's a genetic disease. So, or we can work at the messenger RNA level. That means when the DNA uh, is transcribed and the same language of a basis is used to make a messenger RNA copy, we can work at this level also, or we can work uh, beyond when the messenger RNA is translated into the language of proteins. So the level of DNA, we know that already the adeno-associated virus-based delivery system for gene is really is already in use for various diseases, and it's the most popular system. And even for GNE myopathy, we have groups in the USA and Israel who are trying really hard to uh, use this technology to deliver a normal GNE gene copy into patients. At the messenger RNA level, at least to my knowledge, we don't have any efforts yet in GNE myopathy. But messenger RNA has become, this technology has really matured, especially during the COVID vaccine messenger RNA. We know that it was really helpful to very rapidly produce COVID vaccines. So this technology can be utilized for us also. But beyond that, as we just heard, excellent talk, we have the supplementation possibility with metabolic products of normal protein like sialic acid or the 6-silane lactose or N-acetylmanosamine or the Mannac 6-phosphate prodrug. So all these possibilities are now kind of maturing. We can see that already we have, excuse me, from Japan and Korea, we already have very good data, and so on. I'm sure we'll also get from the Mannac study and then from the prodrug. Now, as I said, all therapeutic options have some limitations. So the adeno-associated viral vector, excuse me, uh, it is a very useful way and is being used a lot, but it has limitations. So the major one being that it's a viral-based delivery system, and the viral generates antibodies once it enters the body. So it is difficult to uh, give a repeated administration of this therapy. So basically, it's a single shot therapy. Because it is single shot, so the dose control becomes difficult, because if you give too much of a dose, then it can be toxic, and if you give too low, then it will be ineffective. And because it is single shot, so you cannot correct uh, once again by giving another dose. The production cost is very high, and also because it's a DNA based technology, so there's a chance of genotoxicity. That means that there could be changes in your DNA. So mRNA based therapy kind of circumvents some of these, and now the mRNA technology has become really good, and so the modified bases are used to make the mRNA, and these are not immunogenic or much less immunogenic. So it's possible to give repeated administration of mRNA. And since you're not altering the DNA, so it's not genotoxic. And now the thing is that mRNA, the way it's designed in the body, in the cell, it is transient, it is unstable. So so it doesn't last for too long, and that's how nature has designed it, and it gets degraded. So the advantage of that is that you can control the dosing. So if you gave too high a dose one time, you can reduce it, and vice versa. But the disadvantage is that you cannot work with a single dose. It needs repeated administration. So mRNA-based therapy has basically two components: the mRNA itself and the vehicle which is going to deliver the mRNA to the cell. That is a lipid nanoparticle. So both of these have been optimized very well, and still optimization is going on. So every part of the mRNA, the different components, I don't have to go in detail. They have been optimized uh, so that the mRNA will translate efficiently into the protein, and it will not be immunogenic, and so to allow repeat administration. And since it's a transient, it's a unstable molecule. So attempts have been made to increase the stability so that you can generate more protein from every molecule of RNA. Similarly, the vehicle which is going to carry the mRNA, the lipid nanoparticle, has also been optimized by changing the lipid composition. And this has been done so that it will avoid the immune attack, and also that it will deliver the mRNA cargo to the right location in the cytoplasm so that the mRNA doesn't get degraded immediately. And also, because we have to give repeated administration, so if if the lipid accumulates too much in the body, it can be toxic. So therefore, biodegradable lipids are used. So the limitations of mRNA therapy is that you need repeated administration, and the translation efficiency, that means how much protein are you making from one mRNA molecule. That so far we have basically used it for vaccines where you need a very small amount of protein, just in microgram amounts. But when we have to treat a genetic disorder, then we need milligram to gram amounts of protein to see an effect. So that is something which still has to be done. Then there's potential toxicity, because if the it has to be repeatedly given, and if the accumulation of the LNP, it can be toxic, and it goes very efficiently to liver, but the delivery to non-liver targets like muscle that remains a challenge. So going to the liver is sort of a natural process because everything goes first to the liver and then the liver sorts it out and removes the toxins. But we wanted to go to the muscle. So that means we have to use a high dose, and uh, problem will come that it can cause liver toxicity because when you have sufficient dose going into the muscle, you have pumped in so much of molecules that it can cause toxicity to the liver. So those are the challenges. But overall, mRNA has advantages. As I just reiterate once again, it's not genotoxic. Dosing can be controlled. Delivery is simpler because you only need a chemically made lipid nanoparticle, not a virus. And the production is simpler and cheaper than AAV vector. So with these in mind, what we are doing at WWGM is to pursue mRNA therapy for GNE myopathy in collaboration with the Tata Institute of Genetics and Society in Bengaluru. So the way we are doing it is, we don't have funding enough to start a lab of our own. So the model we are using is that we are identifying people who are already doing this for something else, and we are making them interested in GNE myopathy. So we are fortunate that at the TIGS, this institute, we have been able to interest them in GNE myopathy, and they've initiated work to try to make mRNA for GNE and to look at the expression and so on. And we also have assurance from Genova Biopharmaceuticals, a company in India, that has produced India's first thermostable mRNA-based COVID vaccine. So they have a platform for mRNA-based expression, and but it needs to be scaled up. So once we have preclinical data, then they have said that they'll be able to do the scaling up for us. So that's for mRNA therapy. Then we believe that the mRNA therapy would be, of course, would even be curative, but is going to take a lot of time, and there are many ifs and buts. The technology. So simultaneously, we should be using some other approaches also, and so we wanted to look at small molecule drugs, which small molecule drugs would be easier to produce and faster to produce. And generally, the cost of production is much lower than nucleic acid-based therapies, and they can be taken orally. So you don't have to go to the hospital repeatedly for injections. So with that in mind, we wanted to look at small molecule drugs for GNE myopathy. And when we look at the literature, we find that one of the pathways that is affected in GNE myopathy muscle is that of autophagy. And what is autophagy? It's a natural process in which the cell kind of does its own spring cleaning. It clears up all the junk, the stuff which has accumulated in the cell, which is non-functional. It breaks it down and uses that to make new components which are useful. So if autophagy process is somehow hindered, then uh, it can be bad for the normal cell functioning, and it can lead to disease. So we found, as I will just show, that various studies are pointing to the fact that autophagy may be affected in GNE myopathy, and that could be the problem why we are getting loss of muscle function. So the first study is from Jason Doe's group in USA, and what he has done is to make induced pluripotent stem cells, iPSCs, from patient uh, blood or skin samples. And then, so this induced pluripotent stem cell, then he has taken it through the differentiation pathway. So he has made it into the muscle progenitor cell, and then further into the myoblast, and then into the myotubes, which then makes the muscle fiber. So what he has shown here in this developmental pathway that the GNE patient iPSCs, they go along the pathway quite okay till the myoblast stage, but after that, they don't differentiate very well into the myotube, and that's why the muscle is lost. So if you look at here, between myoblast and myotube, what exactly is changing in GNE myopathy? So he finds that autophagy is one of the pathways that is very adversely affected. So they they even used they treated these cells with autophagy activator, and they found that it actually does help in the completing the differentiation pathway for the GNE cells. Another study which came from China is here they've studied the fibroblasts from patients directly, not making iPSC, and they showed that here the function of, again, autophagy defect, the function of autophagosomes fusing with lysosome is blocked in the GNE cells. And very interestingly, they show that metformin, which is used very, very commonly in treating type 2 diabetes, this drug actually is an autophagy inducer, and it can upregulate the expression of autophagy, and that can influence the growth and viability of the GNA cells. And the third study, which has come from Korea, this shows, so here they have made iPSCs, not from patients, but they have introduced GNE mutation in normal iPSCs. And then they have compared these iPSCs with the normal ones. And again, they find that the autophagy defects are there. And they are testing a library of compounds in which they are finding some candidates which could reverse the defect. So with this, we feel that we also would like to contribute to this by screening for molecules which could modify autophagy in GNE myopathy. And for this, we are collaborating with scientists who specialize in autophagy research. So this is the Indian Institute of Science in Bengaluru. As I said, that these are people who are already working in a particular area, and now we are able to make them interested in coming and working on GNE myopathy. The last thing that we are doing is to make a natural history study of disease progression in India, which so far is lacking. And we know that it's extremely important to know about, to quantify the disease progression in our own cohort of patients, so that whenever a drug is available, then we have the data. And this, in a slowly progressing disease, doing a natural history can take a long time. So we are now initiating a natural history study with the Indian patients in collaboration with the National Institute of Mental Health and Neurological Sciences in Bengaluru, NIMHANS. Oh, the acknowledgement slide, sorry, it's missing. So I'd like to acknowledge uh, all the scientists and the clinicians who are working with us. So we have three teams who have agreed to work with us. One is this one in NIMHANS, Bengaluru, who are going to do the natural history study. The other one for mRNA therapy is also in Bengaluru, the Tata Institute of Genetics and Society. And the third one for autophagy drugs is also in Bengaluru. Uh, so the good thing is that they're all located in the same city, so they can share, patient samples and reagents and so on. So we hope that these are, as I said, baby steps. We are just beginning, but we hope that in the years to come, we'll be able to make a contribution to find some new treatment options for GNE myopathy. And I thank you all for your attention.